Inhibiting microbial growth in vitro. Control of microbial growth in vitro means to inhibit or prevent growth of microorganisms outside the human body by killing microorganisms or by inhibiting the growth of microorganisms with the use of some methods. The methods in controlling microbes. First is sterilization. All microbes, including cells, spores, and viruses, are completely destroyed. It refers to the absence of microbiological life in anything. Dry heat, autoclaving, gas, various chemicals, and some types of radiation can all be used to sterilize items. Next is disinfection. It is the physical or chemical destruction or removal of pathogens from non-living objects. Pasteurization is a disinfection procedure for liquids. Disinfectants are a type of chemical that is used to kill pathogens. Sanitation is a decrease of microbial populations to level considered safe by public health standards. Antiseptics are treatments for disinfecting skin and other living issues. Microbicidal agents are disinfectants to kill microbes. Bactericidal agents are disinfectants that kill bacteria but not necessarily bacterial endospores. Sporicidal agents to kill bacterial endospores. Algicidal agents are used to kill algae in swimming pools and hot tubs. Virucidal agents to destroy viruses. Pseudomonicidal agents kill Pseudomonas species, and tuberculocidal agents kill M. tuberculosis. Microbistatic agent. A microbistatic agent is a medicine or chemical that prevents bacteria from growing and reproducing. Sepsis refers to the presence of pathogens in blood or tissues. Asepsis means the absence of pathogens. Antisepsis is the prevention of infection, an antiseptic technique, or it is developed by Joseph Lister in 1867 that refers to the use of antiseptics, example washing hands with antiseptic soap. Sterile technique. This technique is a practice to exclude all microorganisms from a particular area so that the area will be sterile. Example or proper technique in wearing sterile gloves. Next is the using of physical methods to inhibit microbial growth. The physical methods commonly used in hospitals, clinics, and laboratories include heat, the combination of heat and pressure, desiccation, radiation, sonic disruption, and filtration. Heat. It is the most convenient, effective, and cost-effective way of sterilizing inanimate objects and materials that can resist high temperatures. Dry heat. Metals, glassware, certain powders, oils, and waxes can be all sterilized by dry heat, baking in a thermostatically controlled oven. Next is moist heat. This heat delivered in the presence of moisture such as boiling or steaming, much faster and effective than dry heat, and may be achieved at a lower temperature, making it less damaging to many materials that would be damaged at higher degrees. Cold. Refrigeration merely slows the growth of most microorganisms. It does not completely inhibit the growth. Desiccation, the process of being thoroughly dried by absorbing moisture. In the laboratory, a vacuum desiccator is a glass or plastic container that aids in keeping materials dry. Radiation. Radiation in various forms, from high-energy radiation to sunlight, can be used to kill microbes or inhibit their growth. Ionizing radiation includes X-rays, gamma rays, and high-energy electron. Filtration. It is the process of filtering or separating cells, big viruses, bacteria, and other microbes from the liquids or gases in which they are suspended using filters of varying pore sizes. Gaseous atmosphere. Microorganisms can be stopped from growing by changing the environment in which they live. Common gases used for sterilization include ethylene oxide or EO, ozone, mixed oxides of nitrogen, and chlorine dioxide. The using chemical methods to inhibit microbial growth. Chemical disinfections refers to the use of chemical agents to inhibit the growth of pathogens, either temporarily or permanently. Disinfectant. A disinfectant is an agent used to disinfect inanimate objects but generally too toxic to use on human tissues. For example, is Lysol. Next is antiseptic. An antiseptic is an agent that kills or inhibits growth of microbes but is safe to use on human tissue. Sanitizer. A sanitizer is an agent that reduces microbial numbers to a safe level. Various factors affect the efficiency of effectiveness of a disinfectant. The bioburden, meaning the type and level of microbial contamination. The contact time means the amount of time that the disinfectant must remain in contact with the organisms in order to kill them, prior cleaning of the object or surface to be disinfected. The organic load that is not present, meaning the presence of organic matter and the materials being treated. The physical nature of object being disinfected, the temperature and pH, the concentration of the disinfectant. Characteristics of an ideal chemical antimicrobial agent. It should kill a wide variety of microorganisms. It should be fast acting, contact time should be short, should not be affected by the presence of organic material, must be non-toxic to human tissues and non-corrosive and non-destructive to materials. It must be soluble in water and easy to apply. Should be inexpensive and easy to prepare. Should be odorless. It must be stable both as a concentrate and as a working dilution. That's all.